Hello, good morning students. Today we'll talk about flow cytometry. Flow cytometry, it is a technique used to detect and measure physical and chemical characteristics of a population of cells and particles. In this process, a sample containing cells or particles is suspended in a fluid and injected into a flow cytometer instrument. The sample is focused to ideally flow one cell at a time through the laser beam, where the light scattered is characteristic to the cells and their components. The cells are often labeled with fluorescent markers, so light is absorbed and then emitted in a band of wavelengths. Tens of thousands of cells can be quickly examined and the data gathered are processed by the computer. So what is there in the process of flow cytometry? That is, the cells they are added. The cells they are of different types. They are labeled differently with the fluorescent marker, and then when they move down, only single cell can cross from the nozzle and the laser beam is put on it and the fluorescence is emitted from the cells and it is detected by the computer and analyzed. So fluorocytometry is used for cell counting, cell sorting, determining cell characteristics and function, detecting microorganisms, biomarker detection, protein engineering detection, diagnosis of health disorders such as blood cancers. Other instrument using flow cytometry includes cell sorter, which physically separate and thereby purify cells of interest based on their optical properties. So what happens? The sample is added, right? Then the cells they are made to pass through a nozzle one by one. Then what happens? The detection plates they are there with positive and negative charges. And the cells they are made to cross them, and depending upon the charge on them they will be sorted to different vials. So in this way cell sorting is there. Flow cytometry is standard laser based technology that is used in the detection and measurement of physical and chemical characteristics of cells or particles in heterogeneous fluid mixture. The use of flow cytometry has increased over the year as it provides a rapid analysis of multiple characteristics, both qualitative and quantitative, of the cells. The properties that can be measured by this process include the particle size, granularity or internal complexity, and fluorescence intensity. These characteristics are determined using an optical to electronic coupling system that detects the cell based on lasers scattered by the cells. A flow cytometer, despite its name, does not necessarily deals with the cell. It deals with cells quite often, but also deals with chromosomes or molecules or many other particles that can be suspended in the fluid. So this detector, the laser beam, right, which is responsible for when the laser beam falls on these particles or the molecules or the cell, the detector it will detect it and gives the information to the computer. So it is helpful for cell analysis as well as the cell sorting. So the basic principle of flow cytometry is based on the measurement of light scattered by particles and the fluorescence observed when these particles are placed in stream through a laser beam. Light scattering results when a particle deflects incident laser light. The extent to which this happens depends upon the physical properties of the particle, namely the size and internal complexity. Forward scattering light, that is FSC, is proportional to cell surface area or size of the cell. Second, side scattering light. It indicates the cell granularity and internal complexity of the cells. The measurement of FSC and side scattering complexity is responsible for detection. The fluorescent marker used to detect the expression of cellular molecules such as proteins or nucleic acid in the system. The fluorescent compound absorbs light energy over a range of wavelengths which is characteristics of that very compound. This absorption of light causes electrons in the fluorescent compound to be raised to higher energy level. The excited electron quickly decays to its ground state, emitting an excess energy in the form of fluorescence, which is then collected by the detectors. In a mixed population of cells, different fluorochromes can be used to distinguish separate subpopulations. The fluorescence pattern of each subpopulation combined with FSC and SSC data can be used to identify which cells are present in the sample and to count that relative percentage. So here the calculation of different types of cells in the blood, that is how many RBCs they are present, WBCs they are present, which type of WBCs they are present, the lymphocytes, T lymphocytes, B lymphocytes, it can be done with the help of the flow cytometry. I hope the concept of flow cytometry is clear. Thank you.